right, everybody. Today we are going to talk about cells. And we're going to talk about the organelles within cells. And we're going to talk about how cells move and how things move in and out of our cells. So we're going to talk about cells and transport. All right. First of all, cells are the simplest structure, and they are the functional unit of living things. I think you already know that. There are about 75 trillion cells in the human body. That is a lot. I mean, really think about that. 75 trillion. And many different types. We have many different types of cells, and many cells are specialized. And that means that they do certain jobs for the body that only they can do, right? You have liver cells that function differently than your brain cells. And you have stomach cells that function differently than your big toe cells, right? So cells are highly specialized. Okay. Cells have a certain structure. The cells within animals are what we call eukaryotic cells. Simple cells are called prokaryotic, and we find those guys in bacterias. Okay? But we are talking mainly about animal cells because we are in the human body. So these are eukaryotic. They have a lot of different organelles and a lot of things going on. One thing all cells have is a plasma membrane. And this is like an outer casing. This basically is the outer boundary of the cell, and it protects the cell and determines what can get in and get out of the cell. It's kind of like a plastic wrap. It is water soluble, and it regulates and is a barrier to the outside world. It is composed of phospholipids. These are molecules that are hydrophilic and hydrophobic, and they orient in a certain way, and they form a barrier and allow certain things in and certain things out. And there are also cholesterol molecules in our cell membranes, in the plasma membrane. And these, remember, cholesterol is a type of fat, and it kind of allows the cell, the plasma membrane and the cell itself to not be too rigid. It allows it to kind of be flexible because flat is kind of mushy, right? So basically this cholesterol allows the plasma membrane to be kind of fluid and to kind of move and not be super rigid. Okay? Here is a picture of the plasma membrane. As you can see, there is what we call a phospholipid bilayer, meaning two. So basically what's happening here is the heads of those phospholipids are hydrophilic, the tails are hydrophobic, meaning water hating. So the tails all get in the middle and the heads go to either side because on either side there is basically liquid. So the tails are hiding in the middle because they are water hating. And then you see some cholesterols, you see some carbohydrate chains, you see some proteins stuck in there. Those proteins deal with channeling items in and out of the cell. Everything is playing a role, okay? So this is just a snapshot of what a cell membrane might look like and all the things that help encompass what it actually is. Proteins are attached or embedded in the phospholipid layers. I just said that. And they are basically passageways for transport of substances in and out of the cell. Depending on what is trying to get in or out, it may need to use a channel or a transport protein because it might be too big to just zip in and out of the phospholipid bilayer. We also see receptors for enzymes and hormones, and there are also carbohydrates, which can serve as markers, or they can deal with cell-to-cell communication. We say that the plasma membrane is selectively permeable, and that means it literally picks and chooses what gets in and out. So depending on what the substance or molecule is trying to get in or out, that plasma membrane is going to say yes or no. It is selective. It is not just letting any old thing in or out. Okay? So there are different things that can help determine whether you're going to get in or out. Okay? And that could be molecular size, solubility, a charge, an ionic charge, or whether there is an attachment to carrier molecules. So your cells are doing so much. I mean, just as you're listening to this, you don't even realize how much your body is performing right now, right? Your cells are making millions and trillions of decisions every moment of every day. All cells have something called cytoplasm, and this is the substance inside the cell. It's a fluid or gel-like substance that fills the interior of the cell. It is mostly water, 75 to 90%, and then there are some other items within the cytoplasm. It is semi-transparent, it is thicker than water, and it is supported by the intracellular membranes and something we call the cytoskeleton, which is one of the organelles within our cells that kind of keeps the cell from collapsing on itself. Organelles are these tiny little guys that live in the cytoplasm, and each one of them performs a specific role. They are distinguished by their size, shape, structure, and function. And now we are going to go through some of those organelles. You should be aware of what the organelle's main function is. Okay? You don't have to know every single thing about that organelle, but if I name an organelle, you should know the job it performs in the cell. First of all, we have the nucleus. This is the largest organelle. It's kind of the brain of the cell. It's where your DNA is housed. And it is enclosed in what we call a nuclear envelope. And this separates it from the cytoplasm. So the nucleus actually has two membranes from the outside world, meaning it has the plasma membrane that encloses just the cell, and then it has its own nuclear envelope that encloses itself. It has pores. This nuclear envelope has pores that aid in movement of materials in and out of the nucleus, because the nucleus does communicate with the cytoplasm, and anything that's going in or out has to go through these pores that are found in the nuclear envelope. Like I mentioned, it contains the DNA. It contains our chromosomes. Humans have 46 chromosomes, 23 pairs. 22 are what we call autosomes. One pair is what we call sex chromosomes, but you have 46 total. And your DNA contains the instructions that control cell function in you. And it contains these in what's called chromatin. There is also what's called a nuclei, which contains RNA and protein and is the site of ribosome production. Ribosomes are made up of ribosomal RNA and protein. Ribosomes are the site of protein synthesis. Ribosomes make proteins, and they can be found singularly throughout the cell or in clusters. There is something called the endoplasmic reticulum. Big word, sometimes we call it the ER. These are membranes that extend from the nucleus throughout the cytoplasm. Their functions are to support the cytoplasm, and they contain channels to facilitate movement of materials within the cell. There are two types of ER, rough and smooth. Rough ER is kind of studded with ribosomes, And the rough ER is the site of where proteins are made and where they are exported from. Smooth ER does not have any ribosomes, and this is where we find lipid synthesis. So rough ER is protein synthesis. Smooth ER is lipid synthesis. We have something called a Golgi complex. This is a stack of flattened membranous sacs. And basically, the Golgi complex synthesizes substances and puts them into vesicles, secretory vesicles. So things, sometimes you think of the Golgi complex as like the packaging plant. It's compiling all these things together and putting things into vesicles. You also have what's called a mitochondria. The mitochondria is called the powerhouse, and this is the site of aerobic respiration. This is where we make ATP. It has an inner fold of a membrane. It has an extra outer folded membrane. And the thought is that mitochondria at once might have been their own cells. And they've actually migrated into eukaryotic cells. You've probably heard of mitochondria. The thing to remember is this is the site of ATP synthesis. This is what is dealing with cellular respiration. If you are in really good shape and you run a lot, your cells might have more mitochondria than most people because you are effective at cellular respiration and aerobic respiration because you're a runner. Then we have something called lysosomes. These are little sac-like things that basically have lots of powerful digestive enzymes, and they help in digesting things. They can digest bacteria. They can kind of ingest cell parts that need to be replaced, or they can ingest like damaged or worn out cells. They're kind of like little stomachs that are just processing and breaking down anything that your cell doesn't need anymore. Here's that cytoskeleton that I mentioned earlier. It is composed of microtubules and microfilaments, which are basically just different types of tubules that give support to the cell and allow for movement of these organelles within that cytoplasm. Because everything's kind of moving around and doing what it needs to do. But if it didn't have any filaments or tubules, it would almost collapse in on itself. So this is kind of like a scaffolding within the cytoplasm that allows organelles to move around and allows the cell to maintain some type of shape. Centrioles are short cylinders of microtubules that are near the nucleus, and they deal with the organization of spindle fibers during cell division. So when your cells multiply through mitosis, we see the centrioles coming out and allowing for this spindle to form where the chromosomes line up and then divide in half and whatnot, and that is mitosis. That is a whole nother lecture, but that centrioles deal with cell division. And then we have cilia and flagella, and these are two types of items that are used in movement. So cilia is numerous short like hair productions that contain microtubules, and they help move substances along the cell surface. Then some cells, such as sperm, have what is called a flagella, and this is a long whip-like production that contains microtubules and basically is almost like a tail, and it enables the movement of that cell. Microvilli, these are extensions of the plasma membrane. They increase the cell surface area and therefore they aid in absorption of substances into the cells. So they are found in intestinal cells, because in your intestines, that is where you start to absorb nutrients. So as I mentioned earlier, depending on the type of cell, it may have a different function. Depending on the function, it may have more of a certain type of organelle. So here's an example of that. Intestinal cells will have a lot of microvilli because you want as much surface area as possible in the intestines, because you want to absorb as much nutrients from the food you're taking in. Okay, now we're going to talk about transport, basically in and out of that plasma membrane. We already said it was selectively permeable, meaning only some things get in, some things get out. And now we're going to talk about the different ways that things do that. So there is something called passive transport and active transport. Passive transport means you don't need any energy. It will just happen. The cell doesn't really have to do anything. In active transport, we need ATP. We need adenosine triphosphate. We need energy to make this transport happen. It doesn't just go. Passive or active. Diffusion is a type of passive transport. Diffusion happens when the net movement of molecules from one area of higher concentration move to an area of lower concentration. It occurs in gases and liquids due to constant random motion of molecules. It occurs in both living and non-living systems. Let me give you an example. I spray some perfume 10 feet away from you. Those perfume molecules are very highly concentrated right where I sprayed them, right? But in time, you will smell that perfume, because those molecules are going to dissipate. They are going to move through the air all by themselves, passively, and they are going to diffuse over to you. And they will continue to do so until they are spread out equally within the room where we are. This could happen in water. If you have a glass of water and you take a drop of food coloring and you drop it in, it's going to drop as like a little ball of color, right? Let's say it's blue. And it's cool. It's fun to watch. You've probably done it. You'll see the little ball drop, and it'll still be really concentrated. But if you walked away and came back in 10 minutes, you wouldn't see that little ball anymore, right? You would just see a glass of blue water, because the molecules have diffused to equilibrium, meaning they went from a high concentration spot to maybe at the bottom of that beaker or glass, whatever you used, and they filled up everywhere. They basically equalized. And you didn't have to do anything. You don't have to stir. You don't have to heat it. You don't have to do anything. They will diffuse by themselves, because things move from high concentration to low concentration automatically. That's just how it works. Osmosis. You've probably heard of this word. This is diffusion of water through a selectively permeable membrane. So this is talking about the cell. Here's that term, selectively permeable membrane. That's the plasma membrane. And we are talking about basically water moving from an area of higher concentration to lower concentration through this membrane, through the plasma membrane. Osmosis. There are different types of solution: hypotonic and hypertonic. Hypo, not enough. Hyper, too much. In a hypotonic solution, we find that a hypotonic solution has a lower concentration of solutes and higher concentration of water. In a hypertonic solution, there is a higher concentration of solute and lower concentration of water. Isotonic means it's equal. So let's think about this in terms of salt water. Let's say we have some salt. That's our solute. In a hypotonic solution, we are going to have less salt in the solution and more water than what's going on outside of that solution, outside of that cell. And it's just the opposite for hypertonic. So here's a picture to kind of show you what I mean. Now we're going to talk about a red blood cell. Okay? So if we drop a red blood cell into a hypotonic solution, there is going to be a net gain of water by the cell, causing it to swell. Water is going to go into the cell to try to make everything equal, right? It's everything wants to be equal. So in the isotonic solution here, this middle guy, nothing's happening because it is equal. The amount of water going in is the same as the amount of water going out. And then the third one is a hypertonic solution, which means there is going to be a loss of water from the cell. Water is going to move out, causing the cell to crenate, or basically shrink. Whereas in that hypotonic, it's going to swell so much that it could actually burst. And that has to do with the movement of water. Everything wants to equalize. If there is more water outside the cell, like in a hypotonic, the water's going to move in. If there is more water inside the cell, it's going to move out. And that's going to make the cell shrink. Those are all passive transports. You don't have to do anything for that. But sometimes our body does need energy to make things transport, and that is called active transport. And active transport requires the cell to use up some ATP. And there are three basic mechanisms: carrier proteins, endocytosis, and exocytosis. Carrier proteins are located in the plasma membrane. We saw those. And they have the ability to bind to a molecule or an ion and basically push it or pump it through that plasma membrane. But doing that uses ATP. And the reason it does is because it's going against the concentration gradient. We're not going from high to low like we like. We're going from low to high. And that's hard to do. That's not natural. You're going against the grain. And in order to do that, you need energy because you're kind of pushing it through. It's not just, going with the flow. Excuse me. Then we have endocytosis and exocytosis. And this is how materials, large materials, are transported in and out. These are things that are too big to go through those carrier proteins. So they're actually going to kind of be put into little vesicles and just kind of popped in or out of the plasma membrane. Endocytosis deals with engulfment of particles and liquid droplets. So literally, the plasma membrane kind of reaches out and makes a little vesicle and pulls something in. Phagocytosis deals with solids, and pinocytosis deals with liquids. But endo, meaning inside, endocytosis means something is being brought into the cell via vesicle. Exocytosis is just the opposite, exo out. So exocytosis is basically some little vesicle moving over to the cell membrane, the plasma membrane, and just becoming part of it and kind of popping out whatever was in it, whether it's liquid or solid. We don't differentiate those names. Endo bringing something in, exo pushing something out. Here's a little picture of an endocytosis, where the plasma membrane is literally just surrounding and internalizing the substance and bringing it in. And it would literally be the opposite of this if it was exocytosis. All right, so we've covered quite a bit of information today with the cell. The cell is, just full of stuff. We just learned that, all those organelles. There's so much going on. So please let me know if you have any questions or concerns, and take care.